Good morning, and what an exciting morning it is. Bonjour. That's the extent of what I'm going to. I can understand you if you speak French, but I'm going to stick today to English, if I have your permission to do so. It is really so exciting to be here, because we have prominent scientists who have arrived from near and from far to come hear about, I think, the wonderful things that they and many of us are working on for hearing health. And so before I actually go into introducing our laureate and our speaker for the day, I'd like to tell you a little bit, if you will, about the Fondation Pour l'audition. This is really a remarkable foundation that started a few years ago. It is why we're all gathered here today. And let me, if I may, show you a few slides and just give you a little bit of background. It's very important for us at the Foundation for all of you to be familiar with what our events, our plans are, and really all the work that's being done for hearing health. So I think many of you know me. My name is Karen Abraham. I am Professor of Genetics and Vice Dean at the Sackler Faculty of Medicine at Tel Aviv University. But here my really wonderful hat is being Chair of the Scientific Committee for the Fondation Pour l'audition. So of course this is not an audience that I need to convince just how critical hearing health is in order for us to maintain. Deafness is recognized as a major public issue by the World Health Organization, impacting about 5% of the global population. Just recently, in March 2018, they came out with the announcement that there are now 466 million people who have a debilitating hearing loss. This is really very dramatic. It also includes 34 million children. And it's predicted that close to, if not 1 billion people will be impacted by 2050. That's not that far away. 1 in 3 people over 65 have a debilitating hearing loss, and of course it gets even worse as we increase in age. And as other medical technology is improving, our population is getting older, and it becomes so critical to help save their hearing so that we can avoid problems of isolation, of depression. And in 2018, there are 1 billion young people who are at risk due to the recreational exposure that some of us have been doing for a long time. And what is the biggest impact, other than health, of course, is that untreated hearing loss costs about 750 billion euros worldwide. Now in France, the problem isn't any better. There are about 6 million French people who have a moderate or severe hearing loss, with 15% of the population having hearing difficulties or tinnitus. And for any of you who have suffered from tinnitus, my husband does, you know that it can be absolutely horrible. Approximately 2,000 newborns are diagnosed with hearing loss every year. And one of the biggest issues that we're trying to help overcome is that hearing loss often remains taboo, particularly in specific populations. It can be misunderstood, and it's very difficult to manage. So many of you are familiar with these details, but what is the Fondation Pour l'audition trying to do in order to help overcome all of these problems? We are really the first public interest foundation focusing on hearing health, and the mission is to engage talents to advanced hearing health, not only to improve everyday life, but also to support research, to support everyday life for people, and to support innovation. And not only have we been able to recruit remarkable scientists, researchers, clinicians, health professionals, administrators, philanthropists to be part of this big project, but we've also been able to, through the generosity of the Belén Foundation, to provide a significant amount of funding. And I don't think that there's anything, if you look throughout the world, even in the United States, there's some major centers for hearing, there is nowhere that has been able to implement the different parts of hearing health as the foundation has done and is going to continue to do. And let me elaborate on those. So first, let me tell you briefly about the scientific committee and for the processing of grants and the scientific prize. And I think most of the members of the International Scientific Committee are here. Can you stand so everyone will know who you are? Thank you. Okay, this is really an incredible group of scientists researchers, clinicians from around the world who really give so much of their time to make this all happen. And so it's myself who's the chair, Dr. Bastian Epp, Professor Adrian Ashraghi, Professor Sophia Kramer, Dr. Frank Lin, Professor Larry Lustig, Professor Brigitte Malgrange, Professor David Moore, Dr. Jonathan Peel, and Professor Christophe Vincent from France. Now we have many types of activities, and I'm going to go very briefly through some of them, and this is important because we would like you to disseminate as much of this information as possible. We're always looking for clients. We're always looking for people to apply for our grants because our goal is to try to help the research not only in France but in the rest of the world as much as possible to get as strong as possible so that we can move to the translational phase of being able to provide solutions for the millions, hundreds of millions of people with hearing problems in the world. We have master's studentships, we have PhD studentships, and these are French residents, can be also in speech therapy or audiology, can be MDs and PhDs. We have postdoctoral fellowships. These can be French who are moving abroad to do a postdoc, or they can be foreigners, international postdocs who would like to come to France to a laboratory. We have MD research fellowships, and this is particularly important to convey because we're very eager for as many clinicians to be involved as possible in the research in the foundation and beyond. And of course, many of you know that we have a call for research laboratories. This is the main part that the scientific committee does. The next call will be very soon in December 2018. We provide up to 300,000 euros per project running for 3 years. We have already disseminated 38 of these project proposals, and the research that's been coming out of the funding that we've been able to provide has really been astounding. And you can see through this list that we really are very broad in our scope. We recognize that multi- and interdisciplinary research is so critical. So of course, basic research, medical and audiological research, applied research and technological innovation, psychology and sociology, because the impact on the people is so critical. We also have, as of course you know, the scientific prizes, and this is part of what our celebration is today. We have the Scientific Grand Prize that for this year is the first time that it was opened to the international community, and we also have early career scientific prizes. So the early career scientific prizes are in 2 categories. We have a category for clinical research as well as one for basic research. And here also we encourage you to tell your colleagues it is not self-nomination. You do have to nominate others, but we believe this is particularly important so that the scientists and clinicians in our community recognize those that are beginning their career and can help them move along. We have the Scientific Grand Prize, of course, which we'll hear much more about further today, and the next call, again, will be in March. And now it's really a pleasure for me to tell you about the early scientific prizes in clinical research. And Dr. Yang Nguyen, can you stand up, please? Where are you? Congratulations. So happy for you. And Dr. Jean-Julien Acouturier, sorry, the name. And for someone, you should know that there is an ad hoc committee that is appointed by the scientific committee. It's approved by the board of the foundation. But for someone who was involved in the process and saw the recommendations, I can tell you that you are really doing remarkable work, and we're so happy to be able to provide this prize to you. And our Scientific Grand Prize goes to Professor Anna Belén Elgoyhen, who I'll tell you about. And I encourage all of you who are here in the audience, and of course our prize winners, to return to Paris when we're going to have the opening of the Hearing Institute that is run by Professor Christine Petit. Christine, come here. I think everybody knows Christine, who has received so many prizes, but most recently the Kavli Award in Neuroscience. Congratulations, Christine. And so please, we encourage you to come and to collaborate and work with our scientists at the Hearing Institute. So without further ado, I'm going to introduce Belén Elgoyhen, and it's such a personal pleasure to do so because I have been following your career for so many years. So let me tell you a little bit about Belén. So Belén was trained in Argentina, and she has, from the start, received so many prizes. In fact, it was such a pleasure to speak to her son, you want to wave, who's here, who told me that he remembers when in 2008, he was young then, quite young, still very young, won the L'Oreal UNESCO Award for Women in Science for Latin America, and I think that this is really such a nice complete circle that now you're getting the prize from the Betancourt Foundation and the Fondation Pour l'audition. And she continued to get prizes. The Outstanding Scientific Personality of the City of Buenos Aires. She received the Bernardo José Achievement Award from the Argentinian Science Ministry. She then won the CONEX Award in Basic Biomedical Sciences, Researcher of the Year of Argentina. She is now a member of the World Academy of Sciences, a member of the Latin American Academy of Sciences, and she was also funded and a Howard Hughes Fellow for many years. And this is really an incredible honor. They're very rare to be given outside of the United States, and really, what a remarkable achievement. Today, Belén is what they call a superior scientist at the Institute for Genetic Engineering and Molecular Biology. She's also the vice director for the institute there. She's an adjunct professor in the Department of Otolaryngology, Head and Neck Surgery at Johns Hopkins, who has a connection also, of course, with the United States. And she was the president of the Argentine Society for Neuroscience. Now, her work has really been very broad because it not only has implications for hearing, but also for neuroscience and for pharmacological sciences. And she was nominated for her seminal contributions to decipher the molecular basis of the afferent control of hearing, discovering the molecular structure of the receptors for the acetylcholine expressed by the cochlear hair cells. Now, we are an audience that are familiar with the cholinergic subunits and their importance, and really the reason that we now understand that is because of Belén's work, and she'll tell you about that and probably her other work as well. So Belén, please come up. I'm looking forward to hearing your talk. Thank you very much. Yes, congratulations. Really wonderful.
So nervous to be here and happy to be here. It's a pleasure, and I really thank Karen and all the committee, the scientific committee and the foundation for giving this award to me. But I will give all the thanks tonight during the ceremony. But since you mentioned that I was here in Paris 10 years ago receiving the L'Oreal UNESCO for Women in Science prize, when I won this prize, and I told the people in my lab and people at the institute, especially the director, he said, "Well, is this prize for women?" I said, "No, it's not for women. It's for worldwide it's a worldwide recognition for people doing science, auditory science." So I'm really happy and I'm really honored. And I'm really happy to be in the Collège de France, which is the heart of so many important discoveries and the heart of science and intellectually. So during my talk today, I'm going to give a sort of an overview of the main discovery for which I was honored with this prize. So it's going to be really about basic science, about molecules, but in the lab we also do work from the bench to the bedside, trying to give people who are hearing impaired in Argentina knowledge about the genetic basis of their hearing loss. So we're doing lots of work in genetics of hearing loss for our population, and it is Viviana Dalamon, who's sitting there, who's come to accompany me here, who's doing that job. Because we really believe that we are privileged in doing science, and we do it to understand the basics of nature, but we have to give the society something back. And since we are we are specialized in the biology of the inner ear, and nobody was doing helping these people who are hearing impaired and who their hearing impairment is due to genetic origin. Nobody was helping them in Argentina. We started with the molecular biology of genetics almost 15 years ago when Viviana joined the lab. So the title of the talk is The Ear Listens to the Brain, the Afferent Olivocochlear System. Can everybody hear me? Yes? And I'm going to structure the talk probably in like 3 modules. The first one is going to be about the molecular cloning of these new genes. The second part is going to be about the molecular evolution of the inner ear and of these molecules, and finally to the physiology that these molecules are involved in. Yeah? So to this audience, probably this we all know. The inner ear is a sense organ who is involved in connecting us with all the surrounding sounds and allows us to communicate with each other. And this is brought about in mammals by a very specialized organ which is localized in the inner ear, here the cochlea, which contains the sensory epithelium for the sense of hearing, which is the organ of Cordy. Now, in mammals, this organ of Cordy is formed by different types of cells. The ones we are going to talk more about today are these ones, the inner hair cells and the outer hair cells, which have different functions in the mammalian auditory system. We all know that the inner hair cells are the phonoreceptor cells in charge of the transduction of sound pressures, which impacts the bones of the inner ear and produces movements of the inner ear. Hi, Frank. Of the inner ear fluids, and in this apex of the hair cells, the mechanotransduction channel, which finally has been identified, or we believe has been identified recently, produces transduction of mechanical information into electrical information that then can be transmitted to the central nervous system by the auditory nerve. And if we look at the mechanisms in the hair cells, the movement of the inner ear fluids produces the shear of the tectorial membrane, the movement of the cilius at the apex of the hair cells, and opening of the mechanotransduction channel, entrance of potassium and calcium, depolarization of the inner hair cell, which on its turn produces the release of neurotransmitter at the base of the hair cells, depolarization and activation of the auditory nerve, action potential, who then sends the information to the central nervous system. And from early on, and from the work of von Bekkesee, who won the Nobel Prize in Physiology and Medicine, we know that the cochlea, if we open the cochlea and stretch it out, it is tonotopically arranged so that different frequencies are detected in different parts of this basilar membrane of the cochlea. Yeah? Now, von Bekkesee was working in cadaveric patients, so he never thought about an active process taking part within the cochlea. However, the fine-tuning of the auditory fibers does not really correlate it to the traveling wave that von Bekkesee had described. And that was because he was working in cadaveric tissues. And early on in the 20th century and the end of the 19th century, Gould was the first one to to postulate that there should be an active mechanism within this auditory system that produced feedback energy into the cochlea in order to fine-tune this basilar membrane. And later on, Rode and Johnston were the first ones to really measure the fine-tuning of this basilar membrane in living organisms. And when the animal died, this basilar membrane turned to be not that finely tuned as von Beckersey had discovered early on. So they were, Johnston and Rode were the first one that could really detect an active process within the inner ear. And if we look at the passive wave, it looks like this, but with an active system that imparts active movement within the inner ear, the amplification of sound is produced, and a very, very fine tuning of every region of this basilar membrane is produced. Kemp was then the first one to record the sounds that were produced within the inner ear, and those are now known as autoacoustic emissions, and that was the first recording of an active process within the inner ear. But the question still remained, what was the basis of this active process in mammals? And it was Bill Brownell who first saw what he then called somatic electromotility. And he was recording in a sabbatical in Danielle Bertrand's lab, and when they put an electrode to outer hair cells and depolarized the hair cells, they saw that the hair cells shrunk, and when they hyperpolarized the hair cells extended. And here we can see this, the hair cell moving, contracting and elongating in response to changes in the membrane potential. So Bill Brownell was the first one to see this electromotility and propose that these changes in length of the outer hair cells was the basis for the active process within the inner ear that produced two things: amplifications of incoming sound and fine-tuning of the basilar membrane. Of course, the question was now, what is the basis for the somatic electromotility? And how is this produced? Now we have the outer hair cells. The movement of the tectorial membrane also produces the opening of the mechanotransduction channels in the apex of the hair cells, depolarization of the hair cells. And this depolarization of hair cells produces elongation and shortening of the outer hair cells. And this is, of course, transmitted to the entire organ of Cordy, and this is thought to be the basis of of amplification. It was years later that the Dallas lab discovered prestin as the molecule involved in responsible for somatic electromotility. This prestin is the fifth member of this SLC26 family of anion transporters. Prestin has lost its transporter function, and what happens is when that prestin can produce intramolecular rearrangements in the response to changes in the membrane potential. And since prestin is highly packed in the lateral wall of this outer hair cells, movement in intramolecular rearrangements of this protein that is highly packed is thought to produce the movement of these outer hair cells. And here we see prestin highly packed in the lateral wall of the outer hair cells. And I took this from one of Christine Petit's paper and Assis' paper and Said's paper. Thank you for that. Beautiful paper. Now, We know now that there's an active process taking place within the inner ear, and this active process is based on somatic electromotility, on rearrangements of presting, and that this produces fine-tuning of the basilar membrane and amplification of incoming sounds. If we look now at the innervation of the organ of Cordy, we see in red the afferent fibers. Those are the fibers that send the information from the organ of Cordy upwards, feed forward to the central nervous system. And we see that the inner hair cells are innervated mainly by type 1 afferents in a very specialized synapse, which has this ribbon specialization known as the ribbon synapse. And then we see that the outer hair cells are also innervated by afferent fibers here in red, which send information to the central nervous system, the type 2 afferent fibers. Now we know lots about this type 1 afferent fibers. We know less about this type 2 afferent fibers, the functions of those type 2 afferent fibers. And there's a very nice work from the Paul Fuchs lab who's starting to decipher the function of these fibers. Now, what is very interesting about the inner ear compared to any other sense organ like the eyes or the sense of smell is that the central nervous system feeds back information to the inner ear. And this feedback information not only reaches to relays in the different parts of the central nervous system, but it goes all the way back to the cochlea here in green, both to the inner hair cell region and to the outer hair cell region. And this is known as the Olivocochlear system. There are two branches of the Olivocochlear system, the lateral Olivocochlear system, which synapses at the afferent dendrites, and the function of this lateral Olivocochlear system is still a matter of debate. We know very little about it. But then we have the medial Olivocochlear system, which makes direct synaptic contact at the base of these outer hair cells. And this is very profuse, and it is it is there in every row of the outer hair cell and the whole turns of the cochlea. This Olivocochlear system was first described in the 1950s by Rasmussen. He did a very nice work showing that this system existed. Later on, it was shown that this system is cholinergic, so that the main neurotransmitter released at the synapse between medial Olivocochlear cells and outer hair cells is acetylcholine. But the main question was, what is the receptor present at that synapse? And that's where our work comes into place. So it has been known for a long time that this synapse between inner hair cells and type 1 afferents is produced by the release of glutamate, which activates postsynaptic AMPA receptor in this very specialized synapse, which has this specialization, which is known as the ribbon synapse. Now, if we blow up this synapse now between medial olivocochlear fibers, which descend from the brainstem and make synapse with outer hair cells, and if we blow up this synapse, what was known until our work came into place was that acetylcholine is released when this fiber is stimulated, and that this acetylcholine activated postsynaptic receptors. However, the nature of these postsynaptic receptors remained unknown until 1994. And there was a huge debate in the literature whether this receptor belonged to those families of receptor which are metabotropic, that is, they are coupled to G protein receptors, which in the case of acetylcholine, it would have been a muscarinic type of receptor. And there was another line of investigation, line of people who proposed that the receptor was ionotropic, that is an ion channel, similar to the nicotinic type receptors, which would permeate either sodium, calcium, or potassium. Paul Fuchs and Murrow working in the bacillar papilla, and also Housley and Ashmore working in guinea pig, and later Dulon working also in outer hair cells of guinea pig, were the first ones to see responses of hair cells to acetylcholine. So what they did, they just isolated hair cells, put them under a microscope, and through patch clamp recordings were able to record the first responses to acetylcholine. And this is taken from chick hair cells from the Fuchs and Murrow paper, I think it was 1992. So in the current clamp mode, if they put ACH, they saw first a depolarization followed by a huge hyperpolarization of the hair cells. Now, in the voltage clamp mode, looking at the currents, they first saw, in response to acetylcholine, an inward current that was subsequently followed by a huge outward current that really was overwhelming compared to this current. And when he applied BAPTA to the solution, in order to chelate all the intracellular calcium, what they saw in the presence of BAPTA, that all this outward current was gone, and they only saw an inward current. So they postulated that somehow acetylcholine was activating a receptor, that this receptor was producing an increase in intracellular calcium, and for some reason this intracellular calcium was activating an outward current, probably carried by potassium ions. And if we look at the papers at that time, and this is 1992 from Fuchs and Murrow paper, the title of that paper was, they proposed a novel cholinergic receptor that mediates inhibition of cochlear hair cells. And I really like to take some quotes from the paper because they are really interesting. The hair cell ACH receptor is neither nicotinic nor muscarinic by the usual pharmacological criteria, because this receptor was not activated neither by nicotine nor by muscarine. It was only activated by acetylcholine. It is equally sensitive to curare and atropine, being curare a nicotinic antagonist and being atropine a muscarinic antagonist. So it had a mixed pharmacology, and is slightly more so sensitive to strychnine, a glycinergic antagonist. More telling still, it is activated by neither nicotine nor muscarine, although carbachol is affected. So here in this phrase, Paul Fuchs and Murrow were describing this baroque pharmacological properties of this unique receptor that's present in the outer hair cells, because it responds to acetylcholine, but it doesn't respond neither to nicotine nor to muscarine, the canonical agonist of nicotinic or muscarinic receptors. It's not blocked by curarity, it's not blocked by atropine, and even more strange, it's blocked by strychnine. Strychnine is the classical antagonist of glycinergic receptor. So they said that the pharmacology of this receptor argues against its being identified with any of known neurotransmitter receptor types. And they said that the hair cell nicotinic receptor may represent a unique class of inhibitory cholinergic receptors. And in a way, they were right in that. And if we look at the papers that came out during those years, this is the Fuchs and Murrow paper. Then we had Omori and Shihimoto postulating that the receptor is a muscarinic coupled to G protein coupled receptors. And then finally, years later, Didier Dulon postulating it was a nicotinic receptor. So that was the state of the art when I came into the picture. And I came into this picture just by chance. During that time, in 1991, these were published in 1992, I was doing a postdoc at the Salk Institute for Biological Studies, this beautiful center of knowledge. And I was doing a postdoc with this very interesting mentor who was Steve Heinemann, who passed away sadly passed away a couple of years ago. And I was surrounded, it was a very exciting time at the Heinemann lab because they had just finished cloning all the glutamate receptors. They had, Holman had cloned GLUR1, Benny Bettler and others had cloned all the kainate and AMPA receptors. And they were starting the technology for making knockout mice, recombinant technology for making knockout mice was just starting. So the Heinemann lab were shifting from cloning to making knockout mice for all the AMPA and kainate receptors. And they were also chasing for the NMDA receptor, who had not been cloned at that time. And unluckily, they were scooped by the Seaberg lab. So nobody was really working on nicotinic receptors in the lab at that time. And there were two people that were very important for my work. One was Jim Bolter, who was a staff scientist in Steve's lab, and the other one was Doug Vetter, Doug Vetter had worked in the auditory system with Muniaini before. So at that time, I was working in nicotinic receptors and using the alpha-7 nicotinic receptor, which was the last nicotinic receptor which had been cloned, searching for an alpha-8 nicotinic receptor. Yeah? And using this low stringing hybridization technique, I came upon a genomic clone which was clearly a novel nicotinic receptor. And it was a genomic clone that was clearly a nicotinic receptor, but was not expressed in the brain. And using in situ hybridization in embryos, we saw that this receptor, this new nicotinic receptor, was expressed in the olfactory epithelium, not in the neuronal epithelium, but in the nasal part of the epithelium, who has the ciliary epithelium in charge of moving the mucus away from the hair cells. It was also expressed in the pituitary. So we went on, and from a library, an olfactory epithelium library, I cloned this gene. And once I cloned this gene, which was clearly within the nicotinic family of receptors, I started expressing this receptor in this heterologous expression system, which is the Xenopus oocyte system, which is a classical system to study the properties of all ion channels. So I cloned this new gene, I expressed it in oocytes, and since it belonged to the nicotinic family, I started doing experiments for probably 2 weeks, putting to the oocytes nicotine, and never got a response with nicotine. So one day I said, well, instead of using nicotine, I'm going to try with acetylcholine. And this was the first time where I saw a response of this injected oocyte. Now, this looks awkward, and when I was preparing this talk with my new students, and I showed them these traces, they said, what is this? I tell, this is how we recorded before. This is a polygraph paper. When I was doing my postdoc, we not only had a computer, but we recorded in polygraph paper. What is a polygraph paper? Well, this is a polygraph paper. And this was the night of the 9/17/93, and this is 1 millimeter. This 1 millimeter is 2 nanoamps, and this is oocyte 1. I put nicotine, 100 micromolar, see no response, but then I put 100 micromolar acetylcholine, and I saw a very little response, probably 2 nanoamp response. But this was reproducible. I washed out acetylcholine and then no response, put acetylcholine again, and there was a response. Wash, no response. Acetylcholine, a very small response. And moreover, this was concentration dependent. So really, this was forming a receptor that responds to an agonist. The agonist has to produce a concentration response curve, and now we have 30 micromolar acetylcholine, 100 micromolar, 300 micromolar, and 1,000 micromolar acetylcholine. So this was a receptor that responded to acetylcholine. It belonged to the nicotinic type family of receptors, but it didn't respond to nicotine. So then what I did is went back to the literature and started reading. And when I started reading and went to the literature, I discovered all those papers from the inner ear, from the Fuchs lab and Murrow and Ashmore labs and Didier Dulon's lab, who were chasing for this receptor and who were postulating a new receptor. And immediately I said, well, this has to be the receptor. It doesn't respond to nicotine. It does respond to acetylcholine. So this was pure serendipity. And this is how science mostly works. I mean, some important discoveries come from serendipity. And this is one thing, now I'm put a bracket here, that we need to teach the ones that are responsible, the governments who are responsible for putting money into basic science, that translational science is not magic, that good science comes from good basic science, and serendipity is part of it. So, of course, once I realized that the receptor was the receptor in the inner ear cell, I did lots of pharmacology. The receptor is this receptor was blocked by nicotine. It was blocked by muscarine, exactly as the pharmacology described by Fuchs and Murrow and Ashmore. It is blocked by strychnine, the glycinergic receptor. It was blocked by curare, and it was blocked by atropine. It really matched the pharmacology of the hair cell receptor. So what we did next was to see if this receptor was expressed in hair cells in the inner ear, and we did both RT-PCR, qPCR did not exist at that time. This is no DNA, this is olfactory epithelium DNA, this is cochlear DNA, and this is sciatic nerve. This receptor is not expressed in the brain, but if we look at all turns of the cochlea, it is expressed in the outer and in the inner hair cell region. So clearly that was a receptor that was expressed in hair cells and who people had been looking for a long time. The receptor is, the hair cell receptor is a ligand-gated ion channel. It belongs to the pentameric family of ligand-gated ion channels, who does not only include nicotinic receptors, but glycinergic receptors, GABA receptors, and serotonin type 3 receptors. It is a transmembrane protein with a pentameric assembly. And since the last receptor that had been cloned within the nicotinic family had been alpha-8 in birds, we named this receptor the alpha-9 nicotinic receptor, an acetylcholine receptor with novel pharmacological properties, which is expressed in hair cells. And I think that this paper in Cell was the first time, I might be wrong, but I think it's the first time that the auditory field appeared in Cell. Now, after cloning alpha-9, although it had exactly the same pharmacology as the hair cell receptor, there were some biophysical properties of the receptor that remained at odds within, when compared to the hair cell receptor. The desensitization kinetics was different, the voltage, the IV curve was different, and the modulation by extracellular calcium was different. And around 1999 and 2000, I was in maternity leave in my house, and I couldn't sleep because this little guy didn't allow me to sleep at night. And I was searching through EST databases, and I immediately realized that there was an EST in gene bank whose sequence was quite similar to that of alpha-9, but clearly it was not alpha-9. So immediately, me and Jim Bolter, who we were collaborating, I was back in Argentina now, realized that there was a new gene that was very similar to alpha-9, and I immediately thought, well, it's the partner that alpha-9 is missing to really fulfill all the criteria to be the receptor in hair cells. So we went in and cloned. Now we had built a hair cell library. We cloned the receptor, a new receptor, and named this receptor alpha-10. We subcloned the gene in BlueScript and started to express this receptor in all sites. And since this new clone, this new gene was found over my sleepless night because of this guy, which was like that at that age, we named this clone in BlueScript that we were using P. Bruno 1.0. So P. Bruno 1.0 is alpha-10 in BlueScript, which we have used, and I have sent over to all labs wanted to use the clone. P. Bruno, Bruno is the name, of course, of my son. No? So after cloning this, we started doing in situ hybridizations. Alpha-10 is expressed in the outer hair cell region, in the inner hair cell region, in embryonic tissue. It is expressed in adults in the outer hair cell region. It's not expressed in the inner hair cell region, so it's developmentally regulated, and this makes sense because there is a developmental regulation of efferent innervation that I'm not going to go into now because I don't have the time, and alpha-10 and also alpha-9 is expressed in vestibular hair cells. And moreover, if we now looked at the biophysical properties of the recombinant alpha-9 and alpha-10 receptor, it completely resembles the properties of the native receptors of hair cells. So it is highly permeable to calcium. If we look at the voltage IV curve, current voltage response of alpha-9, we see an outward rectifying current, and alpha-9, alpha-10 has a completely different voltage IV curve, which really resembles that of her cells. Alpha-9 and alpha-10 heteromeric receptors are biphasically modulated by extracellular calcium. Responses are small at 0 calcium. They get bigger at millimolar, low millimolar calcium, and then it's blocked. Whereas alpha-9 is only blocked, but it's not potentiated by calcium. And alpha-9, alpha-10 receptors desensitize, while alpha-9 receptors do not desensitize. So clearly, alpha-9, alpha-10, recombinant alpha-9, alpha-10 receptors, the properties of the recombinant receptors really match those of the hair cell receptors that had been looking for. And then we published this alpha-10 receptor in 2001 at the same time as Larry Lustig and Paul Fuchs also had cloned this receptor and published the sequence of the receptor and the expression of the receptor, but I think there was no function in that paper, functional characterization in that paper. So with the cloning of alpha-9 and alpha-10, we have closed the cloning of all the members of the nicotinic family of receptors, which is now divided into the muscle type receptors, which are present in the neuromuscular junction, which produces the muscle contraction. Then we have the neuronal type receptors, which go all from alpha-2 to alpha-8, and it includes beta receptors. And then we have the hair cells receptors. Hair cells only express alpha-9 and alpha-10 receptors. Now, of course, there are numerous things that are different between alpha-9 and alpha-10 and all other members of the nicotinic family of receptors. And we knew that from the beginning because at front, it does not respond to nicotine. So there has to be something different in the sequence of that receptor. And if we look at the sequence, the homology between alpha-9 and all other nicotinic subunits, we see that the homology is very, very, the identity, amino acid identity is very, very low. It's below 40% all along, except the homology between alpha-9 and alpha-10 with 60%. So this means that alpha-9, alpha-10 are members of that family, but they are distant members of that family, very distant members of that family. And for that, probably they don't respond to nicotine, because there must have been different changes in the amino acid sequence which led these receptors to be unresponsive to nicotine. So at that time, we had cloned the receptors. We knew how to express them heterologously in synaptozoocytes, but we were very interested in jumping into doing some electrophysiology in native receptors. And at the same time that we had cloned this, Elizabeth Glowaski in Paul Fuchs' lab had established the cochlear explant ex vivo preparation that was very easily now to do electrophysiological recordings from hair cells, much easier than in isolated hair cells. So we started a long-lasting collaboration which still lasts till today, and Eleonora Katz in the lab, who started with me almost from the beginning, and she is sitting there, went several times to Paul's lab and collaborated with Elizabeth and brought back to Argentina the cochlear explant. And we started doing electrophysiological recordings in cochlear explants, and these are typical responses from inner hair cells, and we see, if we put acetylcholine, we see an inward current followed by the outward current. And we started doing comparisons and lots of work with the heterologous receptors we had cloned and the in vivo or ex vivo native receptors with the cochlear preparation. The first thing we did was an enormous comparison of the pharmacological properties, and as I told you, the receptor responds to acetylcholine, but it doesn't respond to any other agonist. Most drugs we know of are antagonists of this receptor. Nicotine is an antagonist, curare is an antagonist, atropine and muscarine are an antagonist, but what is more awkward is that strychnine is a very potent antagonist. Again, strychnine being the classical canonical antagonist of glycinergic receptors. Bicuculline is also a potent antagonist, and bicuculline is the canonical antagonist of GABAergic receptors, GABA-A receptors. And this drug, ICS, is also a very potent antagonist. So it is, and ICS is the canonical antagonist of 5-HT3 receptors. So we had cloned a receptor which was a distant member of the nicotinic family, and it shared pharmacological properties with members of the other members of the family, with GABA receptors, with serotonin receptors, and glycinergic receptors. Moreover, the receptor was highly calcium permeable, and the ratio of calcium to monovalent permeability is around 10, so that is really, really high. It is as high as the NMDA receptor, who mediates synaptic transmission in the central nervous system. And moreover, we were able to determine the stoichiometry of this receptor, and this was done by Paola Plazas in the lab. The hair cell receptor is a pentameric receptor assembled from 2 alpha-9 subunits and 3 alpha-10 subunits. So finally, over the years, we could decipher the molecular constituents of this synapse. We know that the medial olivocochlear system releases acetylcholine, acetylcholine activates alpha-9, alpha-10 receptors, which is highly permeable to calcium. Calcium goes in and it activates a nearby SK2 potassium receptor, which is highly sensitive to calcium. Potassium goes out from the cell, and this produces hyperpolarization of this hair cell. This first inward current is due to the entry of sodium and calcium through the alpha-9, alpha-10 receptor. This outward current is due to the efflux of potassium, and we were not the ones to discover this SK2. This was done by other people in the field, but with all this discovery, the molecular constituents of this synapse was now deciphered. So far I've shown you that the hair cell cholinergic receptor is the one alpha-9, it mediates transmission at the MOC, the medial olivocochlear efferent hair cell synapse. It is a nicotinic receptor. It's formed by alpha-9 and alpha-10 subunits with an two alpha-9s and 3 alpha-10 subunits. That's the stoichiometry. It is a distant member of the family, and it is highly calcium permeable. Now I'm going to talk a little bit about the molecular evolution of this receptor. If we look at the evolution of the auditory system, and we go way, way back in the evolution to the early tetrapod amnions, we know that there was a cochlear amplifier there. There were very few cells, hair cells in the basilar papilla, but they had amplification. And amplification in those early amniotes was based in ciliary motility, in the adaptation of the ciliary motility, in the adaptation apparatus. Now, with the branching of the different evolutionary branches, the archosaurs, the lepidosaurs, and the mammals, the tympanic middle ear appeared in parallel in all the in all the species. There was division of labor. Two types of hair cells, which was at most developed in the cochlea of mammals, where now we have outer hair cells for somatic amplification, inner hair cells for phonotransduction. In mammals, then we have the elongation of the coiled cochlea to accommodate more frequencies. The mammals are now the ones that are more sensitive to the highest frequencies in the animal kingdom. We have the appearance now of somatic electromotility based on rearrangement of a protein known as prestin. So with these morphological changes that took place and functional changes that took place in the inner ear of mammals, most likely molecular evolutionary changes took place within molecules. And one gene that really changed, and molecule that really changes function, is prestin. Prestin is present in other species, but it is only in mammals where prestin has suffered evolutionary changes in order to serve as the motor protein. So knowing this, we started looking at the evolutionary history of nicotinic receptors. And this is a phylogenetic tree of all the nicotinic receptor subunits that are known. And there's something very peculiar about this phylogenetic tree. If we look at all the nicotinic subunits, like for example the alpha-2, the alpha-4, the alpha-3, the alpha-6, and we condense the tree from all species to the nearest node, we see that all known alpha-2 species from all vertebrate species can be condensed to one branch. All the alpha-4s to one branch, all the alpha-3s to one branch, and so forth until we reach alpha-9 and alpha-10. And when we reach alpha-9 and alpha-10, we see that all alpha-9 condense together, but they are very similar to non-mammalian alpha-10 subunits. Alpha-9, all alpha-9 condense together, but they're also similar to non-mammalian or nearer to non-mammalian alpha-10 subunits. However, mammalian alpha-10 subunits form a different branch and more distant to the non-mammalian alpha-10 subunits. So this means that for some reason, mammalian alpha-10 subunits have acquired non-synonymous substitutions in the coding region, and they now have diverged from the early alpha-10 subunits. So non-mammalian alpha-10 subunits stayed very near in sequence identity to alpha-9, but mammalian alpha-10s and only mammalian alpha-10 subunits started to acquire non-synonymous substitutions in the coding region of the protein, now forming a different branch. And this was analyzed by Lucia Francini. I'm not going to go into the details of her work, by doing in silico molecular evolutionary analysis based on KS analysis and codon-based likelihood models. She showed here in red that different regions of the alpha-10 in mammals acquired non-synonymous substitutions in the coding region. The protein changed, and it changed in the coding region, and it still functions. So it means that those changes might were conserved along the evolution and probably acquired a novel function only in mammals. And probably the evolutionary history of alpha-9 and alpha-del was the following. They both branched early on from a common ancestor. There was a gene duplication, alpha-9 and alpha-10 appeared. Alpha-9 continued almost the same in all vertebrate species. Alpha-10, non-mammalian alpha-10, continued very near in identity to the alpha-9 vertebrate species, but for some reason, at one point in the evolution, mammalian alpha-10 subunit started to acquire changes in its coding region. Now, if a protein goes through the costly and energetic process of acquired changes in the coding region, it must mean that it acquired a novel function. And this happened only in mammals, and probably this followed the evolutionary history of the auditory system in mammals. Yeah? So then we started asking, what's the difference between mammalian and non-mammalian subunits? I mean, is there a functional difference between receptors formed by mammalian proteins and receptors formed by non-mammalian proteins? And one of the most astonishing differences is that mammalian subunits have a very high calcium permeability. Non-mammalian subunits have a very low calcium permeability. So this is very astonishing, and one starts to think, why do we need a mammalian protein with a high calcium permeability and a non-mammalian protein with a low calcium permeability, since in both systems the responses are the same. You need the entry of calcium through the alpha-9, alpha-10 receptor for the subsequent activation of a potassium channel. And of course, we don't know the answer to this puzzle, but recently the Fuchs lab has shown that in apical cells of mammals, instead of SK potassium channels, there are BK potassium channels. So remember that BK potassium channels have a very low calcium sensitivity. They respond to changes in voltage. And remember that hair cells in mammals have acquired a high frequency sensitivity. Apical hair cells are sensitive to high frequencies. So we can hypothesize that this changed in the permeability of mammalian receptors who acquired a high calcium permeability, most likely accompanied the acquisition of high frequency sensitivity in mammals. Of course, this is a hypothesis. It is only a puzzle that still needs to be resolved. So far, alpha-9, alpha-10 mediates synaptic transmission at the MOC efferent cell synapse. It is a nicotinic receptor. It has an alpha-2, alpha-3 alpha-2, alpha-9, 3, alpha-10 stoichiometry. It is a very distant member of the family, and it is high calcium permeable only in mammals, not in other vertebrates. Now let's shift to the last part of the talk, which is the physiology of the efferent system. We know from early on that the efferent system is an inhibitory system. We knew it was cholinergic, and we knew it is inhibitory. It inhibits amplification, it inhibits the output of auditory efferents. And this is probably one of the first recordings in 1956 from Robert Galambos, who was recording electrical activity from the round window in cats while he was putting click tones, and he was recording these responses at different intensities of the click. In the upper panel, these are different intensities. This first response is probably electrical activity in the hair cells. This N1 response is the electrical activity in the afferent fibers, the auditory nerve. Now, if at the same time, and this is from the original paper, and I really love this. This is after Rasmussen, where Galambos, at the same time he was giving the click to the round window or to the inner ear, he stimulated at the base, at the midpoint in the hindbrain. He was stimulating here the efferent system. And while stimulating the efferent system, the hair cells response was still there, but the N1, that is the auditory response, was completely diminished. So this was the one, let's say, of the first indications that the efferent system is inhibitory, while stimulating at the base of the fourth ventricle, the N1 response, the auditory output, was completely diminished. And moreover, if we look, let's look at the left, at the right panel, and these are different intensities of sound, but let's look at one intensity, for example, 6. And these different curves are different frequencies of stimulation. When the frequency of stimulation was low, 20, the inhibition was very, very low, but as he increased the frequency of stimulation, the inhibition produced by the medial olivocochlear system was enhanced. And this was reproduced by many others, for example, Gifford and Guinan, Galambos working in cats, Art and Fettiplace working in turtle. And this, what we show, what is shown here is inhibition of output of, in this case, the N1 auditory output. This is almost the same as a function of the increase in the frequency of stimulation of the efferent system. So at low frequency of stimulation, the inhibition is very low. But as soon as the frequency of stimulation is enhanced, maximal suppression of auditory activity is obtained. So at this point, Jimena Ballesteros in the lab and Eleonora wanted to decipher what is the cellular basis for this suppression. Why does the efferent system need to respond at high frequency stimulation, while low frequency stimulation is almost inadvertent, is not seen by the hair cells? So what Jimena and Eleonora did was to stimulate the efferent terminals to outer hair cells, and at the same time record from these outer hair cells. So what they're doing, they're stimulating this terminal, releasing vesicles, and activating alpha-9, alpha-10, and SK2 potassium currents, and recording the excitatory postsynaptic currents from these receptors, from these hair cells, in the ex vivo preparation. Now, if the frequency of stimulation is very low, there's lots of failures. You don't see any postsynaptic currents, and here is an excitatory postsynaptic current. But as soon as the frequency of stimulation goes up, from 20 hertz to 25 to 50 to 60 to 80, the inhibition builds up. And this build-up of inhibition with increasing frequency of stimulation is due to the plasticity of this synapse and due and due to two very important phenomena. The first one is presynaptic facilitation. This synapse has a very low probability of release. At very low frequency, it doesn't release. The probability of releasing one vesicle is very low, but as soon as the stimulation increases, calcium builds up and the probability of release goes up. So, and the second mechanism is that postsynaptic facilitation. So since calcium starts building up, the potassium SK2 potassium channel continues open for a longer time. So plasticity at this synapse, due to presynaptic facilitation and postsynaptic and summation, is underlying this increase in efferent responses, efferent suppression, with increase in frequency of activation of the efferent system. And moreover, if one performs a sustained stimulation of the efferent system, which is here sustained for 1 second, the efferent system continues to release vesicles, and the suppression is sustained while the terminals are stimulated. This is looking at current, but here we look at voltage. This is membrane voltage. At high frequency stimulation, you see a sustained hyperpolarization of the hair cell at high frequency stimulation. Now, how can this hair cell sustain, this efferent system sustain stimulation for longer periods of time at high frequency stimulation? If we look at an electron micrograph of the synapse between the efferent system, this side, the outer hair cell, this side, we see that the efferent, MOC efferent has lots of vesicles. It's filled up of vesicles. Here it's a 3D reconstruction done by Paul Fuchs. The efferent system is filled up of vesicles, and that is why it never depleted. It can really fire at very, very high frequency. And this, of course, goes in hand with the physiology of the system. So this is what Jimena found. Jimena found that that postsynaptic inhibition builds up at high frequency. It's very, very. The inhibition is not produced, or postsynaptic inhibition is not produced at low frequency. There is a region where the inhibition builds up and then it saturates, and it is within the firing frequency of the MOC system. And this clearly matches what was described in vivo for the physiology of the system, where responses inhibition is very low at the low frequency of stimulation, and then suppression is maximal at high frequency. So what Jimena has found is probably that the plasticity as this synapse, as the efferent olivocochlear MOC hair cell, outer hair cell synapse, the plasticity at that synapse and the response of that synapse can clearly explain the way the MOC system functions and transfers inhibition to inhibit the gain of the auditory system. So this is the sum of this part. The MOC outer hair cell synapse has a very low probability of release because it has a very low quantum content. The probability of the release increases at high frequency. This is due to presynaptic facilitation and postsynaptic summation. The responses of the MOC outer hair cell synapse mimic the responses in vivo, and therefore the plasticity of the MOC outer hair cell encodes the transfer function from efferent fiber frequency to the gain of the cochlear amplifier. Now, as I told you, the efferent system really works at high frequency. So the higher the frequency, the more the inhibition. And this goes in hand with one of the functions that has been proposed for the efferent system, which is protection from noise-induced trauma. And this is what I'm going to go into this last few minutes. The hypotheses for the MOC function of this efferent function are several. Some have been deciphered, others have not been deciphered. Of course, it controls the gain of the system. It inhibits the gain of the system. It has been proposed that it mediates selective attention, visual versus auditory. When we need to do a different task, when we are reading, for example, we need to reduce the input to our ear, to the auditory system, in order so that we can concentrate on our reading. It controls masking from background noise in order that we can filter noise from our background. It shapes the normal cochlear development, and it protects the inner ear from acoustic injury. And it is here where we have done some work collaborating with Charlie Lieberber's lab. We know that the exposure to loud sounds is one of the causes of hearing impairment. From the ambient causes, not the genetic causes, exposure to loud sound is the main cause that leads to two things. It leads to hearing impairment because we lose hair cells, and once we lose hair cells, so far mammalian hair cells do not degenerate, and therefore once we lose them, they are gone. And this is a typical audiogram showing hearing loss at high frequencies, which is the typical hearing loss due to exposure to high noise levels. But it also, exposure to noise, high traumatizing noise levels produces tinnitus or acouphens, which is the perception of a phantom sound in the absence of an objectivable sound source, and this can be very debilitating for lots of people, and I'm not going to go into that, but we're working on that. Now, over the years, once cloning alpha 9 and alpha 10, we did different genetically modified mice that we have used and shared with the community, and I'm going to talk about two mice, the alpha 9 knockout mouse and an alpha 9 knocking mice. Alpha 9 knockout mice completely lack responses. If you knock out alpha 9, even if you don't knock out alpha 10, responses in hair cells are gone. And not only responses in hair cells are gone, but the physiological output of that is gone, which is the inhibition of distortion product autoacoustic emissions. If you have here a normal level of autoacoustic emissions and you activate the efferent system, the amplitude of autoacoustic emissions diminish. Control stimulation, the amplitude diminished. Control stimulation, the amplitude diminished. This is a wild-type animals. In alpha 9 knock-in animals, inhibition by the efferent system of distortion product autoacoustic emissions are gone. Really, the alpha 9 knock-in animals are completely de-efferented. There's no efferent inhibition going to hair cells. That's one animal model we produced. We produced a second animal model, now not a knockout, but we introduced one point mutation in the transmembrane region of alpha 9, which we call the L9' T mutation because we changed leucine for a threonine in the transmembrane region of the receptor, and that increases the gating kinetics of the receptor. So now the receptor is open for a longer time. There's spontaneous opening of that receptor. The receptor does not desensitize it, so we call that a gain of function mutation. So now we have an efferent system that most likely is going to work more powerful, a more powerful efferent system. Inhibition is going to be more profound. And we use both those animals. These are results from Carolina, who is sitting there. In the knocking animals, we did some physiological recordings to show that we had a gain of function in these animals. If this is a postsynaptic response in outer hair cells of wild-type animals here in black, we see that the knocking animals, although the responses are a little bit, the magnitude is lower, the responses are longer and longer. So the actual charge here that the outer hair cell receives is much longer. Now, if we do stimulation of the efferent system, at high frequency, we see that in wild type, we see responses that go back to baseline, responses that go back to baseline, but in the L9 prime knocking animal, the responses are sustained in time. And if we look at the membrane potential in the current plant mode, we see that the hyperpolarization produced in these animals is really, really profound and sustained compared to wild-type animals. So yes, we have generated an animal who has a gain of function of the MOC system. And if we look at the in vivo physiology of the animals, and this was done by Stéphane Masson in Charles Lieberman lab, and we see inhibition of distortion product acoustic emissions. We see in the inhibition in black is of the wild-type animal when he shocks at the base of the fourth ventricle, and in red is the knocking animal. The inhibition is really, really profound, and it is prolonged over time. So we really obtain a gain of function mutation with an enhanced medial olivocochlear system. And what we looked at now is at the consequences of these mutations, both the knockout mice and the knocking mice, in noise-induced hearing loss. Yeah? And we found that really mice that have an increased gating of the alpha-9 receptor are completely protected from noise-induced hearing loss, and moreover, they are protected from what's now sort of trendy, which is synaptopathy and hidden hearing loss. And this is shown here. So here we Luis Boero and Maria Eugenia Gomez Casati, what they did was to expose these mice to traumatizing noise. In control, in black, we have the control response. This is ABA thresholds as a function of frequency. So in black, we have a control. 1 day after trauma, of course, ABA thresholds go up. But in wild types, 7 days after trauma, the ABA thresholds go down. Yeah, this is a normal response in wild types. Now, if we look at wave 1 amplitude, again, controls in black. 1 day after trauma, the amplitudes really go down, but then they recover. However, they don't recover, they don't overlap the control. So this is typical of what Lieberman and Kurosawa have described as hidden hearing loss. Yeah? Now, what happens in the alpha-9 knocked mouse and in the alpha-9 knocking mice? Again, here we have the control. 1 day after trauma, we have an increase in ABA thresholds, but in the alpha-9 knockout, when there's no medial olivocochlear system, the ABR thresholds never recover. So these animals, in the absence of a functional medial olivocochlear system, are more sensitive to noise-induced hearing loss. And moreover, the recovery of the amplitudes here in gray is not complete, and it's more important than, more pronounced than in wild type. And what is more and more astonishing is what we see in the alpha-9 knock-in, which has an enhanced medial olivocochlear system. First, the ABA thresholds don't go up. They're completely protected against hearing loss, and the amplitudes of the wave 1 amplitude of the ABR are not changed. So really, the amount of damage produced by nose exposure is correlated to the function of the medial olivocochlear system. We can demonstrate this with these 2 animal models. Of course, this is not new. This has been proposed early on by several labs, starting by Rajan and then by Charlie Lieberman and others. So in summary, I've shown you today that in the first part, the cloning of alpha-9 and alpha-10 as the receptor that mediates the synaptic transmission between MOC, medial olivocochlear fibers, and hair cells of the cochlea. I show you a little bit about the molecular evolution and the structure of that receptor. I showed you that the synaptic plasticity at that synapse is really the transfer function between efferent system and modulation of the gain of the auditory system, and that the medial olivocochlear system is key to protect the inner ear from acoustic injury. So finally, the people who really did the work, I, of course, started doing this when I was a postdoc, continued doing this in Argentina after that, but there were lots of people who followed upon that discovery, and it's really them who make things happen in the lab. This, I think I haven't missed anybody, and they're not in order of importance, but in the order that I remember that they came and joined my lab. I won't mention all of them because it would take forever, but I just mentioned the ones that are here. Eleonora Katz, who was with me from probably 3 years after I came back to Argentina. Then Viviana Dalamon, who is sitting there, who is the one working with hearing impaired patients, trying to find genes that produce hearing loss. And then Carolina, no sé dónde estás, Caro? Wedemeyer, Carolina Wedemeyer, who worked with the knocking and did the electrophysiology. And later on tonight, probably Javier Zorrilla de San Martin will be there. He's not here now, but he will be there tonight because he's doing a postdoc here in France. And then Marcela Lipovsek, who did the evolutionary story, will be here tonight. She's coming from London because she's working at King's College London. And of course, all the collaborators, a long-standing collaborator with Fuchs. We still do it today at UCLA with Jim Bolter. I started with Jim Bolter in Steve Heinemann lab. Now he's retired. Doug Vetter in Minnesota, and of course Charlie and Stefan at Harvard Medical and Eaton Peabody lab. Of course, the funding, this could not have been possible without all the funding we have received over the years. Meager funding from Argentina. I mean, the funding from Argentina is very, very small. Those grants are very, very bad, very weak. But of course, over the years, we've been funded by Howard Hughes, NIH, Forward, NOHR, Human Frontiers, Tinnitus Research Initiative, and all this funding from abroad is really what makes things happen in a country like ours. And now, from now onwards, of course, the Fondation Pour. And finally, I think I haven't missed anybody. This is the faces of all those names that were written there in the lab. And thank you for listening to me this morning.